And here we spoke about our total body water and our two fluid compartments, intracellular fluid. We described this as being inside the cell. Extracellular fluid we described as being outside cells. And then to put some values here, we said this is about 50, closer to 70% of our total body water. The extracellular fluid being about 30% or so. And then we further divided the extracellular fluid into the plasma. This we described as being fluid in vessels. And that was a larger chunk of that 30%, right? No, I'm lying. That was a smaller chunk, so that's about 10%. And we also said this had the interstitial fluid, which was the fluid around tissues. So not in vessels, not in cells. And this is a larger chunk of that extracellular fluid. So of that 30%, about 20% or so. Okay, so just to remind us of our fluid compartments and the relative proportions there. The next thing we spoke about was homeostasis. Homeostasis. And for homeostasis, we spoke about the whole loop of events. We talked about the set point, the players such as sensors, detectors, the integrating centers. I'm not going to go through the entire loop of events, but I'm going to talk about the two important concepts that kind of came out of that. This was negative feedback, which were described as changes that correct the error signal. Okay. We also described as moving towards the set point. We gave a whole bunch of examples, such as body temperature, such as blood glucose levels. We even talked about that example with the car and the gas. So make sure you understand that loop of events in terms of the sensors, the receptors, the detectors, the integrating centers, and then make sure you understand the concept of what negative feedback is in relation to all of that. Okay, and some examples. We also spoke about positive feedback. This is more rare. Okay, we said this was very rare, right? Hardly found throughout the body. And we also described it as being a brief period of moving away from the set point. Okay, we also gave some examples for this. We spoke about the uterine contractions. We spoke about blood clotting. So basically moving away from the set point in terms of potentiating a change or potentiating that error signal, but for a brief period of time and for a very specific reason that usually was incorporated in a larger negative feedback, right? So this ended with a negative feedback. Okay. Just to remind ourselves of some examples here, we said clotting, blood clotting, and those uterine contractions. Okay. All right. We next spoke about enzymes. I'll review what we talked about enzymes, some of the important parts here. Okay, we said that enzymes, first of all, they lower the activation energy, but then they also speed up the reaction. Okay, and make sure you understand what that means in terms of the activation energy and how it changes in terms of the way the reaction proceeds. Okay. We also said that enzyme rates can increase, so things that affect the rate of enzyme activity. And we mentioned a few things here. So the first one would be the inherent catalytic activity, right? The catalytic activity of that specific enzyme. We also said the relative concentration, so the substrate concentration, the enzyme concentration. Right? Those two things affected the rate of the enzyme activity. We then said temperature and pH also play an important role. At higher temperatures, at acidic pHs, we have faster rate of activity of enzymes. And then we said the affinity of the substrate for the enzyme, or the enzyme for the substrate. Okay, so these are some of the properties that we said would increase the rate of the enzyme activity. Okay. I also want to point out here two important concepts that came about in terms of our discussion of enzymes. We talked about endergonic reactions, and we spoke about exergonic reactions. Right? It's really important for us to differentiate what these two are. So we said endergonic reactions require energy. Exergonic reactions liberate or donate energy. We also compared endergonic reactions to anabolic reactions. That's another way to say this. Or endothermic is another way to say this as well, endothermic versus exothermic. And then we compared exergonic reactions to being catabolic. Okay. And just to put some examples here, we said that building up amino acids to make proteins was an example of an anabolic reaction or an endergonic reaction. So if you make proteins from amino acids. And then an example of exothermic, exergonic, or catabolic would be to break down proteins to form amino acids. Okay, so make sure you understand the difference between these two and examples. All right. Any questions so far? Okay. The next set of things we spoke about had to do with biomolecules. And we'll go over these briefly here. So we spoke about biomolecules next. And we said these are the main components of living things. Our biomolecules are mainly made of carbon, hydrogen, nitrogen, and oxygen in varying quantities. And we spoke about different types. So we had lipids, carbohydrates, nucleic acids, and proteins. Okay. Most of them were polar, meaning that they played nicely with water, right? They had that uneven charge around the molecule, right? So they interact nicely with water. Interact well with water. Whereas lipids were not, right? Lipids are non-polar. They do not interact with water. One of the important examples we gave here were phospholipids. And we said that they were amphipathic. And these formed what important structure? What important structure do phospholipids help to form? The bilayer, right? The micelle and the phospholipid bilayer, more importantly, okay? So the cell membrane or the phospholipid bilayer. Okay. Okay. All right. Among this topic, we also spoke about things like condensation reaction, We also spoke about hydrolysis reactions, so make sure you understand the difference between those two. We also talked about in proteins the different structures, so primary, secondary, tertiary, quaternary. Make sure you go over those three and differentiate them. Yeah, and I think that was it. Okay, all right. We then spoke about, what is this? Cellular respiration. Okay, basically how our cells can break down biomolecules to harness energy. And we mainly spoke about the context of glucose. Bless you. So we said this is basically how cells can harness energy from biomolecules to synthesize. What do we synthesize when we break down these biomolecules? Let's have currency in the cell. What is the whole goal of breaking down glucose, the whole goal of oxygen? Energy, yeah, but specifically what was the currency? ATP, exactly, perfect. ATP, okay? And with that ATP, we can do cellular work. Okay, so we spoke about the 4 processes involved here. So we'll just kind of go over them here quickly. Glycolysis. We talked about the linking step in between. We talked about the Krebs cycle. And then the most important, the electron transport chain, where oxidative phosphorylation occurs. So we'll mention where each of these processes take place and kind of what's produced, right? Cytosol is where glycolysis takes place. And we know here we make 2 net ATP. Okay, we also produce NADH, which is the reduced form carrying some electrons, but make sure you understand the difference between oxidation and reduction. Right, so NADH, NADH, which goes off to the electron transport chain. We talked about the linking step where pyruvate is used. All right, so the pyruvate that we make up here, let's put the pyruvate. Pyruvate that we make from the glycolysis goes into the mitochondria. Sorry guys, I'm getting ahead of myself. Right, so this happens in the mitochondria. Okay, so cytosol versus mitochondria. And so that pyruvate is shuttled off to the mitochondria, where it's the substrate for that linking step. And what we produce is acetyl-CoA and some carbon dioxide. Okay. The Krebs cycle, we know, also takes place in the mitochondria. Here we use this acetyl-CoA that was produced. Right? It comes down to this Krebs cycle. And what we yield is some more ATP. We also yield some NADH in the reduced form. And then we also yield another carrier that kind of helps with shuttling electrons as well. And then some carbon dioxide. Okay, the electron transport chain is the most important. This is where oxidative phosphorylation takes place. This happens across the inner mitochondrial membrane. What we produce here is 34 net ATP. And this uses oxidative phosphorylation. which we know requires energy. Requires oxygen. Okay, this is where oxygen becomes that final electron acceptor. Okay, actually this shouldn't be here. This is a substrate in this process and not a product. Okay, it's a product up here. Okay. All right, so make sure you understand the difference between oxidative and substrate level. So I'll write up here that we make this via substrate level phosphorylation. And make sure you understand the difference between what that means. Okay. Any questions so far? Any questions so far? I won't go into the cell membrane. What we spoke about was really straightforward cell membrane. We talked about the most important part of the cell membrane being the phospholipids and their amphipathic nature. So make sure you go over that and the other structures in the cell membrane. Really straightforward. So what I'll go into next is the cell membrane transport. So we spoke about the driving forces. Okay, let's just put down some terminology here. We had the membrane potential, we had the equilibrium potential, okay, we had the chemical driving force, the electrical driving force, and let's kind of describe what these things mean. So the membrane potential was due to the uneven distribution of charge, unequal distribution of charge. Across the cell membrane, right? That positive versus negative, with the inside of the cell being more negative with respect to the outside of the cell. We spoke about the equilibrium potential, okay? The equilibrium potential was determined by the concentration gradient. And this was basically the membrane potential at which particle movement is equal to 0. Okay, for example, we said that potassium's equilibrium potential was -94. So at that membrane potential, at -94, there is no movement of potassium because the chemical driving force is equal and opposite to the electrical driving force. Okay, so that was really determined by the concentration gradient. We also spoke about the chemical driving force, which is based on what? What determined the chemical driving force? Not the charge. This is chemical. What determines this? The concentration gradient, right? The concentration gradient. So that high to low concentration is what determined the chemical driving force. Whereas the electrical driving force was determined by two things. What were those two things? So one was the charge, so the charge on the particle. And then something else. We talked about that polarity of the cell, right? The polarity of the cell, the negative versus positive. The polarity of the cell. Okay. All right. Now, in that respect, we also spoke about the electrochemical driving force. And we said that this is basically the sum of both charges, the sum of both the chemical and the electrical. The sum of both forces, I'm sorry. Okay. Oh, I'm so sorry. All right. And so the last thing we talked about with regard to the chemical and electrical driving force is how do we determine the overall movement of particles when we combine these forces? So we said that if both forces were in the same direction, that was nice and easy and straightforward. But if the membrane potential was greater than the equilibrium potential, then the electrochemical driving force would favor which of our two forces? Electrical or chemical? Electrical. We said if the membrane potential was equal to the equilibrium potential, then the ECDF equals 0, right? There's no movement. And then we said if the equilibrium potential was greater than the membrane potential, then the ECDF favors the electrical driving force. Okay. I'm sorry, yes, chemical. Yeah. Thank you. Chemical driving force. And remember, this is only if the forces were in opposite directions. If they're in the same direction, then that's nice and easy and straightforward. They both go in that direction. But if they're in opposite directions, then we have to deduce which force is more dominant. Okay. All right. Now the last thing we spoke about here before we wrapped up was the forms of movement, the forms of cellular transport. Okay. So we talked about active transport, which had two other types, passive transport, which had three types here. Okay, so first of all, we said that passive transport, let's kind of just describe this here, was spontaneous. Another way to say that is a downhill reaction or donates energy. Active transport was non-spontaneous or an uphill reaction or requires energy.